great.
Oh, John, you're muted.
I forgot. There you go. So can you hear me now?
Yes, we can hear you.
Okay, great. Okay, well, thank you, Ashley, for the introduction and for organizing this. And thank you for inviting me to give this presentation. I'd like it to be as informal as possible. So please put questions in the chat box, and I'm happy to interrupt the talk as we go, or save questions to the end, whichever works. So really, it's really hopefully as informal as possible. So without further ado, I shall start talking about sustainable control in gastrointestinal parasites of small ruminants. So what I wanted to do in this talk was blend a little bit of the research we've been doing with some basic information on parasite control and the kind of trends in terms of the way things are changing, both right now, currently, and in the future, where we're trying to head. And hopefully that will become clear as we move through the talk. So these are the parasites we're talking about. This guy here is Homunculus contortus, which is one of the worms, but we're talking about a group of parasitic worms which live in the gastrointestinal tract of both sheep and goats, and both sheep and goats share largely the same groups of parasites of this group. So just a few slides, which are kind of a parasitology refresher for some and maybe an introduction for others. But there's lots of these different parasitic worms, but they can kind of be categorized into two different groups. And so one group is literally one species, and that's Homunculus contortus, and is sometimes also known as the barber's paw-worm, and that's because it feeds on blood in the host, and it causes anemia, essentially. And this can cause very severe disease from literally sudden death through to just severe anemia, right through down the spectrum to just chronic poor doing, fail to grow at the adequate rate for lambs, etc., etc. So it's got a full spectrum of disease, but is often severe. When we look at the other group, this is more complex in the sense there's multiple different species, which are closely related, but do have differences. And they cause very different disease syndrome, and that's diarrhea, mainly of lambs and weaned lambs. So it's the older group of lambs. When you see diarrhea in very young lambs, it won't be this. It's that older group from 4 months onwards that typically you see problems. And so I should go back to Homunculus. That's the blood feeding one. It's important to be clear that doesn't cause diarrhea. So if you see diarrhea in lambs, it's not due to Homunculus. With Homunculus, you see anemia. So we have to think of these groups of parasites quite separately. So again, in terms of a refresher, there's these different species, but they basically all have a similar life cycle. And understanding this is absolutely critical to understanding management. And it's becoming more critical as we'll talk about in terms of having to be a little bit more thoughtful about the sustainability of control practices. So very rapidly, and I'm going to keep this as brief as possible. If we go through the life cycle, I'll just note a few key features which are important. The adult worms live in different parts of the GI tract. They breed. The female worms produce large numbers of eggs which pass out in the feces. And then these hatch and develop into these microscopic larvae, which then develop within the fecal pat into the L3. And then the L3s migrate out onto the pasture. And these are the infective stages. And these get then ingested by the sheep and develop to adult worms. And the time it takes from these L3s being ingested to develop into adults is typically about 3 weeks. Another wrinkle of the life cycle is that some of these parasites can inhibit their development in the immature stages and don't develop to adults. They choose to stay inhibited, and they can stay as these quiescent stages for many, many months. And that's actually how parasites like Homunculus survive over the winter inside the sheep. And that's one of the adaptations they've got to, which were essentially originally tropical parasites, actually Homunculus have adapted to these colder climates. So when we talk about control, and we'll get to this as we go, we're talking about trying to manage both or minimize the accumulation of these infective stages on the pasture, and also the extent to which sheep are going to ingest them, and that's where grazing management comes in. Another thing it's important to emphasize is we monitor these infections with fecal egg counts, and we can, many of you will have done this either yourselves or certainly sent samples off to have it done. And that gives you an overview of the parasite burden, but what it doesn't tell you is which of these species you're dealing with, and that can be a limitation of our monitoring tools. So one of the themes here is parasite control over the last 20, 30 years has been far too focused on really the use of dewormers, the reliance on dewormers to control parasites. And part of this was, the problem has been, these drugs have been really almost too successful for their own good. When they were first launched, they're highly effective. Almost people lost knowledge about how to manage parasites in other ways simply because we just rely on these drugs now. And as I'll show, that one of the problems with this is drug resistance, and that's now threatening the sustainability of the whole system, if you like. So again, I wanted to do a quick refresher. For some of you may know this, some of you less so, but just in terms of what drugs are available, there are two main groups of drugs which we've used for many years in Canada, and that's this group here, Safeguard and Valbazan, which is fenbendazole, albendazole, and these are essentially the same. They're slightly different drugs, but they're essentially the same in terms of the way they act. And then the other group's ivermectin. And what I will tell you is that we've got a lot of resistance problems to these products now. There are less commonly used dewormer classes, although producers here do use them. One is Sidectin, which is moxidectin, and one is Prohibit. So I spelled that wrong on making the slide. Prohibit, and that's levamisole. And these are not licensed here, but can be sourced from the US and through a veterinarian as well. So these drugs we don't have as much resistance to because we have not used them as much, and I'll come back to that. And then there's these two brand new drugs, Flucavar, which is closental, which is a very narrow spectrum drug against Haemonchus contortus and doesn't act on those other worms. What I should have said with these ones, these are quite broad spectrum and act when they're working against all the different species, whereas Flucavar basically only acts against Haemonchus and not the others. And then there's this new drug here, Startect, which is a combination drug, which is, again, very broad spectrum. So these two new drugs licensed in 2016 and 2018 in Canada are really a godsend to the industry because they are still highly effective. But the issue about these, and this is one of the things we want to emphasize, is we really should treat these as precious commodities, because they will go the same way as the other drugs if we don't manage their use properly, and that's part of what I'm going to talk about. So one of the other things in terms of just general things is when we talk about treating and using these dewormers, you've got to remember that basically what they do is they kill the adult worms in the sheep, but then they don't have persistent activity. One of them, Sidectin, has some persistent activity, although when we've got ivermectin resistance, that persistent activity actually goes away. So essentially, the drugs available we have no persistent activity, which means you treat an animal, you kill the worms in it, but from the moment you've treated it, the animal's then getting reinfected again from the pasture. So it's a temporary respite, if you like, once you use one of these drugs. And as I said earlier, it takes about 3 weeks for these ingested L3s to get to adult worms, and then you start to get pasture contamination again. So really, you could remember that these single treatments do something at a moment in time, but you've got to think about the pasture in terms of reinfection, and that gets worse, as I'll explain, as you get later into the grazing season. Another concept I wanted to introduce, and some of you may have heard of this, but I just wanted to spend a little bit of time explaining it because I'll come back to it in more practical senses a little bit later. And there's this concept of refugia. And so refugia is defined as a portion of the parasite population which isn't exposed to the drug when you treat with drug. So when you think about it on a farm, if you think of the farm as an ecosystem, A lot of the parasites are actually on the pasture, depending on what time of year it is. And then the other parasites are actually in the animals. And so when you treat with a drug or a dewormer, the only parasites which are exposed to that drug are the worms inside the sheep that was treated. So all these parasites on the pasture and any parasites which is any animals not treated with the drug are not exposed to it. And so there's no selection for resistance. And therefore, and the term used for that is these parasites are in a refugia. And when we think about using these drugs in a sustainable and effective manner, a lot of the current thinking is about now maximizing this refugia population. And this hopefully will make more sense when I get to it in a practical manner. So these are my diagrams, which I'm very proud of. I made them many years ago, drew them myself, but I'm not an artist, I have to admit that. But this is kind of to try and explain the concept of what refugia is and why it's important. So this kind of diagram shows sheep on a pasture. These letters represent worms, either on the pasture or inside the sheep, and the S's are ones which are susceptible to the drug, and the R's are the ones which are resistant. So in this situation, you've got a lot of parasites on the pasture. So when you treat with drug at that time and you kill the worms inside the host, only the resistant survive. And so only these produce eggs. And so they will pass out onto the pasture, but they're diluted out by all the susceptible parasites on the pasture, which is why resistance occurs gradually, almost unnoticed, until it gets bad enough that it's causing major problems. It's an insidious thing. But if you take a different situation where you've got very few parasites on the pasture, and most of them are in the host, and you treat with drug at this time, then only the resistant parasites survive. They produce eggs onto the pasture. They're not diluted out to anything like the same extent. And so when you've done that, you much more rapidly select for resistance. So the point about this is it's not just the number of drug treatments that we use, it's when we use them, which can be a major factor in how quickly we get resistance problems. And it's quite interesting. I came to Western Canada about 13 years ago, and when we came here, there wasn't really any knowledge of the current situation here. And a lot of people said, well, we don't think we're going to have resistance problems like they do in the US, for example, because we use drugs less often. Therefore, there's less drug use. But we, as I'll show in a minute, we do have resistance problems here. And at least part of the reason for that, I'm sure, is because of this refugia, because in Western Canada, there are times of the year where there's very few worms surviving on the pasture. All the worms are inside the sheep. And so if we treat, for example, use in this situation, this is late winter, early spring, that's a really strong selector for resistance. And so, so that's part of the reason we have resistance here, but also something to think about in terms of when we treat is critical, not just how often we treat. So in various parts of the world, these kind of issues have become more and more to the fore. And this is just examples of information in the UK from the British Veterinary Association about responsible anthelmintic drug use. And that's just gaining traction everywhere in the world to a greater or lesser extent. And obviously where the sheep industry is the biggest tends to be where most of the publicity is around it, but the same principles apply wherever we are. So really, in a nutshell, we want to move from this situation where we really rely on the drugs to do the job to a more integrated parasite control approach. And that I've tried to break down into three components. One is using drugs in a more sustainable manner, which is represented by this segment of this chart. And then the other is grazing management and how we manage grazing to maximize parasite control. And then there are a variety of alternative strategies, some which are kind of being used in, well, they're all being used in different parts of the world, but are things to think about going forward for the industry here. And I'm going to spend more time on these two and then talk about these a little bit at the end, but with the question and answer session, I'm happy to kind of explore any of these individual things as we go. But I want to focus on these two because these are the here and now of what we can do to improve management.
John, there's just a question that came through in the chat box.
Yes, let me have a look. If I use indoor. Oh, good question. Yeah, and that's normally I actually make that point in my talk. So thank you for that question. So the question is, if you have ewes indoors in a heated barn, can you deworm in the winter indoors, or is that a waste of time? Good. It's a very good question. It depends a little bit on the context. First thing to say is there's no transmission indoors of any of these parasites. The ewes will not ingest parasites from bedding. They don't survive in that environment. So there's no infection while they're indoors. So if they've been treated prior to going indoors, there's no point treating again because they will not have acquired new parasites. If they had large parasite burdens in terms of inhibited larvae, you could think about treating in terms of what they'd acquired from the pasture the previous year. If there's reasons to think they're carrying burdens from the previous grazing season, then you might be justified in doing that. So it depends on the context. But any completely indoor systems don't suffer from these parasites. These are transmitted on by grazing. Does that answer the question or more clarification?
No, that's pretty good. We were treating ewes indoors, and basically before we'd wean their lambs off them.
Right.
And then the ewes would go outside to a pen to dry off, and then if it, when summer grazing came around, we'd go out to pasture.
Yeah.
So my thought prior to by deworming in the barn, then they'd shed those parasites and eggs in the bedding pack in there, which would get hauled out and put into a compost pile and not infect the pasture theoretically.
Yeah, I mean, as I said, the only reason you would treat indoors is if there's reason to believe they've still got parasites from when they were last outdoors, if you see what I mean by that, right? And and in terms of what happens after you've treated them, anything that comes out, if you like, the back end of the sheep after treatment will be dead anyway. So that won't contribute to pasture contamination. Yes. So basically, I think does that make sense in terms of what you said, or?
Yeah. No, we've been in pretty good shape as far as parasites go, so that's why we had kind of gotten away from the deworming in the barn, because we've been doing fecal counts and things and not seeing real issues in the ewes, doing Fibonacci scoring and things, and
Great.
And for the most part, everything was looking pretty good. I was just curious if we start to see a problem, if that's kind of a good way to tackle it, or.
Yeah, no, and I think the other thing, there's different elements to this that we can circle back around to at the end if you like. But I think for example, a bad thing to do potentially would be to treat with drug in the barn if they were still carrying parasites because of previous grazing. They're in the barn, you treat them everything with a drug, and then move them onto a pasture. That potentially would be a bad thing to do because you're selecting for resistant parasites, which will then contaminate that pasture. So doing a targeted treatment, if you're going to do it, would be a better way to do it. And I'm going to come on to targeted treatments a little bit later. Yeah. So basically, it's all about thinking what parasites have inside them at that moment in time, and that's going to come from whatever pasture they were last on. They will not have acquired anything from the barn itself. Okay, so in terms of the information resources for this, there are a whole variety, and I've put some up here, and these are all great sites. But particularly if you look at United Kingdom, Australia, it's not directly relevant to here in terms of the detail. A lot of the principles are great, and it's all good. A good site is one produced by Paul Menzies at Guelph, which is a booklet which is available. Again, these things date quite quickly, but it's still broadly relevant. And so that's a good information source. But again, because of geography matters a lot, what we've done is we've produced a new website, which we've just launched, which is specifically for the situation in Western Canada. So, I encourage people to look at that and look at the information on there, which is, as I say, some of it's generic in terms of just general educational things. And other parts of it are very tailored to here, such as the current situation here, the dewormers available, etc., etc. And there's some resources, links to other sites through there. So I'd encourage people to look at that and also send me feedback on it in terms of what's missing or what's not clear, etc. So what I wanted to talk about kind of in the intervening part of this talk is a little bit of an update on a research project we ran. This is myself and Dr. Michelle Levy at UCVM. Camila Quiroz was a graduate student in the group, now as a postdoc in the group. And the way this project ran, it was funded by what is now RDAR, but was Alberta Agriculture. And it basically ran from 2015 to 2019. And we had a whole series of DVM students in our program. There's summer students on the project, and then also some biological sciences students. So one side benefit of this was to get some of our DVM students actually involved in sheep work and working with sheep producers, which I think was a good outcome of this. So, and again, there's people on this call who may have been involved in this project. So thank you for collaborating with us on it if you did so. So there are a variety of objectives to this project. One was a kind of scientific technical exercise about introducing new technologies for identifying parasite species and drug resistance. And then a lot of it was using those tools as a survey to really try and understand the situation here in Western Canada. Because I say, if you go back to pre-2015, there was no empirical data on the situation here. So that was the objectives. And when we, the basic approach was to send out these sampling kits to producers, both pre and post drug treatment, get them to send them in. And then we would look at the parasite burdens before and after drug treatment, and that will tell us something about the parasite populations as well as their susceptibility to the drugs being used and how effective the drug treatments were. And also apply these DNA sequencing assays to give us more information on species and resistance. And then we provided reports back to producers as we went. Just a few things to highlight about the flocks that participated. We have a wide variety of flocks from very large to very small. Whole load of different breeds. It was an eye-opener to me as just how many breeds are in use in Western Canada. The whole variety there. A lot of producers did report having problems with diarrhea to varying extents and land mortality, as you might expect. And then a little bit of grazing information was we found that well over half of the people who responded to the questionnaire that we supplied didn't move lambs after weaning to a new pasture. A few things about drug use. We can see something like well over half producers are treating 2 times or more a year. And then this one's interesting to me that, at that time, which is a few years ago now, most people were treating the whole flock as a block rather than part of the flock. And that's something I wanted to kind of revisit as we go through this talk. And then we produced this technology to provide more information. I don't want to label the technology, but basically we extract parasites out of samples, do DNA sequencing approaches. And what this does, the bottom line is you could extract thousands of larvae from a sample. We can tell the species which are present quantitatively. So the output we get from this is, as well as an egg count from a pooled sample, we can see how that breaks down with the different species of parasite. And also, I'm not going to talk about it here, there's some of the drug resistance mutations that we can identify and directly say whether the parasites are resistant or susceptible to a particular drug. But I don't want to make this a research talk, but I just wanted to introduce the relevant parts. So this is what we found. If you look at this, each one's a different flock. It's just part of the data. You can see just across the top here, the eggs per gram on pooled samples, and this is ewes, mainly in June, early July. And you can see a lot of flocks have very high egg counts and some very low egg counts. And then if you look at the species breakdown, it's very dominated by Homuncus, which is the red one, which is the blood-feeding one. But also we've got these other species in there as well. So different flocks at any moment in time of different issues with different parasites. And that's an important thing to understand, is it's not a homogeneous problem for everybody, both in terms of the extent of it and also the particular parasites that you're dealing with. We also looked at the effectiveness of people's drugs. And I'm not going to present that data of drug treatments. I'll revisit that just tangentially as we go. But we also visited farms and actually tested for resistance properly with a fecal egg count reduction test, where we weigh animals and dose them accurately and look at the response to treatment. And this table here shows the bottom line. When you look at ivermectin, for Homuncus contortus, 11 out of the 13 farms visited had resistance in that parasite to ivermectin, 3 out of 8 to this parasite, which is one of the diarrheal ones, and 1 out of 9 to this one. Similarly with fenbendazole, which is Safeguard, similar kind of results. And levamisole, moxidectin, a lot less resistance. So you can see from this that these two drugs that people have been relying on for many years are pretty much shot for Homuncus, and these other parasites are developing resistance as well. And this is typical of what's seen across the world. What we know, for example, with moxidectin, which is Cydectin, still highly effective, but we know from other parts of the world, once you get ivermectin resistance, this quickly follows if that drug is used. And so if people start switching to Cydectin, it won't be long before we see resistance to that, because it's a similar drug class. And as part of this project, we reported back information to producers to really try and encourage people to think about, are the drugs working? Do they need to choose a different drug? Is there drug treatment needed, even in the first place? What parasites are a problem on their farm? And we've tried to provide that back, working with the veterinarians to provide feedback. And so I think this was successful. We've had a lot of positive feedback. I've certainly enjoyed it a lot. I'm working with the sheep producer community here. That project is now finished because the funding's come to an end, and actually COVID stopped us last year doing on a small scale. We hope to re-engage with not exactly the same project, but other related projects depending on funding going forward. So that's that. So that was really just a snapshot of the research to give you a feel of the current situation, and to really hopefully convince you that the problem is real and not just assumed. And so what I wanted to do now is talk about this, how do we improve sustainability of control? So really moving away from this assuming the drugs work and just doing that, versus now thinking more strategically about how we integrate different components of parasite control to achieve control, but in a more sustainable manner. And I've broken down to these 3 areas, and I'm going to just deal with each in turn. And probably a general comment to make is no one thing is going to work for everybody. No one thing is going to be a complete solution. And obviously what's suitable for somebody with a small flock is very different from someone with a large flock. Somebody's doing some indoor housing or not, et cetera, et cetera. Somebody's got a lot of grazing management options versus not. So you can imagine it's a very bespoke thing about what works on any one farm. So the idea is to be more tailored and thoughtful about the approach. So a lot of it I'm going to talk about is how we use these drugs more effectively and sustainably. And we think about this as drug stewardship, about avoiding overuse or inappropriate use, and moving away from this, let's just treat just to be on the safe side culture, to actually treating when we need to treat and only when we need to treat, and using other approaches where it's all possible. And when we think about doing that, we've got these specific approaches that I'm going to go through one by one, which are about using these things effectively, but minimizing this development of resistance. And again, I'm encouraging questions as we go. If people have got anything I say which isn't clear or need clarification, or just got any general questions. So the first point is monitoring, and I think this is really important. because it's good to know what, some producers will barely need to use drugs. Others will have to still rely on them. And it's about deciding in your flock, but do you really need to or not? And actually on the cattle side, I don't want to go into cattle, a lot of producers use drugs when it's not necessary. I think in sheep, for the foreseeable future, we're going to need them, but in a much more targeted manner. So the idea with monitoring is to sample over the season several times, sampling ewes in the spring when the egg counts are highest, and then ewes and lambs in the summer and or fall. So 2-3 times a year is ideal. And then when you do that, sampling from 15 to 20 animals in a group. Now, you can pool samples together and do an egg count on that pool to give you an overview of the parasite burden in the flock or the group you sample from. If you do that, it's important to use the same amount of feces from every animal to get an accurate view of it. And I know that some producers do fecal egg counts themselves. We did a couple of workshops on that a few years ago. I don't know how many people are still doing it, but some people I do know and other people send off. But if it's done in a pooled manner, it's actually quite doable in terms of cost, etc. And we can combine these with these molecular tests and provide more information I've described, but that has to be done within the research project at present. And this approach can also, with people with more larger flocks and more complex grazing, can really also help assess risk in different groups in different areas of the farm. And then also sampling both pre and post treatment to assess the efficacy of the drug is actually important, because we can't assume that the drug treatment has worked, and hopefully I've shown that. And so I think it's important people are doing that, preferably every year after treatment, taking a sample 2 weeks later and seeing if there's still eggs there or not. And that can again be done in a cost-effective manner. The 2 things about using the drugs is if anybody's got any practical questions on this, just throw them in the chat box about how you take samples, how you store them, all those kind of things. Happy to chat about that as time allows. So when we look at different ways we use the drugs, there's 2 terms that you may have come across. One's targeted treatment and one's targeted selective treatment, sometimes abbreviated to TT and TST. And so with targeted treatment, we're still talking about treating the entire group, but making sure the timing is targeted. So we're treating when it's needed, but also treating when the maximum number of worms are in refusion. And so, so trying to choose that time. Now that's not always practical, but it's something that is talked about, and we can come back to that. And then the other one is targeted selective treatment, where we're just trying to treat those individuals in the group in terms of which ones need to be treated, rather than treating everything. So again, illustrating it with these diagrams, if you look at treating all individuals in the flock and this is a low refusion situation. So there's very few, in the way I've drawn it, parasites on the pasture. And so as I pointed out earlier, ewes in the spring would be like this. And so if you treat, or actually moving from a barn onto a clean pasture, for example, would be another example we talked about earlier. So if you treat the animals in this situation, you treat everything, the only parasites that reproduce and contaminate the pasture are resistant, you get rapid selection of resistance. If you only treat some of the animals, then some susceptible surviving those untreated animals, they contaminate the pasture as well. It slows down the process of resistance by this dilution effect. And so by only treating some of the sheep in the flock or animals in the group, that really helps slow down resistance. And so I've just again used this example of using the spring as an extreme example of that. And so if we feel we need to treat ewes at lambing time, targeting the treatment to some of the ewes, not all of them, is one of the biggest things you can do to prevent resistance occurring. And the question that comes up about that is, well, yeah, that's all very well, but I'm worried I need to make sure I'm covering all those animals at risk, etc., or reduce pasture contamination as much as possible. But the answer to it really is, if you look at the way parasites are distributed in a group of sheep, it's not even. not all animals in the group have the same parasite burden. In fact, it's very skewed. And so this is just a made up data set just to illustrate the point. If you categorized animals by fecal egg count, for example, the vast majority would have very low counts and a few would have very high counts. And so when you think of it like that, it makes no sense to be treating everything the same. It's a waste of using drug even without the selection for resistance in terms of cost of product and time doing it. So really what you want to be able to do is just treat the ones that need it at this end of the spectrum. And so, that's this idea of targeted selection treatment is increasingly gaining traction in different parts of the world. And then the question becomes, what proportion of animals do I treat? What are the numbers we're talking about here? Well, it's approximated that 80% of the worms are in 20 to 30% of the individuals in the group. And so you really should be aiming ideally to be treating no more than a third of the animals in the group. Now that obviously depends on the situation. So that's not a rule, a cast iron rule, but that's an aspiration and an aim. And obviously it could even be less than that depending on the situation. And so again, one trick to convince people to do this is to start, cautiously, and once you're comfortable with it, go further down that spectrum of treating less animals. How do you choose animals to target? Well, it was a great question earlier because they're obviously thinking about these things, which is great. Obviously it has to be practical and quick and easy. And so you can't use fecal egg counts. It's obviously completely impractical at individual animal level unless you've got three individuals. The kind of things you can do is use FAMACHA, which was mentioned in the question, and that's a scoring system of looking at the level of anemia and only treating the animals at this end of the spectrum with anemia. So 3s, 4s, and 5s, depending on the situation. And so leaving those with the pinker mucous membranes to be untreated. And that works well for Homoncus. And there's on the website we've produced, there's a link to how to do that properly. And I encourage people to look at that if you're not familiar with that. But of course, it doesn't cover these other parasites. And so if you've got a mixed parasite situation on your farm, then other things in conjunction with this, such as condition score of ewes, or looking at the nutritional stress on ewes in terms of number of lambs they're carrying, looking at lambs either based on condition, or in a number of places now they're using live weight gains and target weights, and if lambs are not reaching a certain target weight, just treating those which are not reaching the target weight. And I'm just going to show very quickly a few things to convince you, hopefully, that there's some merit in this. It's a difficult thing to prove empirically that it does slow resistance. All the modeling studies that have been done suggest it should, but there's been a number of studies to kind of demonstrate that it does have an effect. And this is just an extreme experiment to make the point. Just look at the green and the blue lines. This is the percentage efficacy of a drug over several years, showing how it's declining. And this has been done, to an extremist with monthly treatments. You can see you can select for drug resistance by repeated treatments. But if you only treat a proportion of the group, that's the TST line, you prevent that resistance occurring. So it is effective. And you think of these new drugs that we have now, if we want this to be the situation and not this, clearly. And then when you look at the actual effects on production, there's been a number of studies now done which tend to show the same results, and that is if you compare daily weight gains of animals with targeted treatments versus treating the whole groups, that there's little or no difference in terms of production benefits. And then there's obviously an economic benefit because you're using less product, spending less time, dosing animals, etc., etc. So if it's done properly, it's a win-win in terms of cost savings, not losing any production benefits or disease risk, not increasing disease risk, and also really maintaining the efficacy of these precious dewormers that we have. The other thing which is important in terms of not selecting resistance is avoiding underdosing. That's a really strong selective resistance. And so I've just listed things which are obvious, really, but often don't get applied in practice. Is using correct technique, making sure drenching guns are calibrated, weighing animals prior to dosing is, if at all possible, is absolutely the way to go. If you're doing it at a group level, dosing based on the heaviest in the group rather than the average, because if you take the average weight and dose everything the same, you're going to underdose some animals. Obviously not using expired products, collect storage of products, and not physically mixing products together. There are times when we use combinations, but that shouldn't be done by mixing products because that changes the activity and uptake of the drug. Another thing to think about is you don't want to bring resistance on a farmer's purchase stock. So we should always think about quarantine dosing, and that's treating sheep that are purchased with drug and keeping for at least 48 hours them off the main pastures. That gives the time for the drug to work. And so there's a bit of a paradox here when you think about it, though, because if we're saying that we've got a problem, we don't want to bring drug-resistant parasites in, but we're using drugs to stop that, it seems like a little bit of a paradox because the drug won't work if they're resistant. And that then comes down to drug choice. So thinking about where the animals are sourced from to inform our drug choice. So in this part of the world, we know that ivermectin and fenbendazole resistance is common. They're not going to be very useful for quarantine dosing. So what we advise is using a combination of anthelmintics. And so, for example, some of these new products or products which are not used commonly here, using in combination, again, sequentially, and don't mix them, is the most guaranteed way you've got of not purchasing resistant parasites into the flock. And then ideally checking 10 to 14 days later whether that has worked and there are no eggs in the feces prior to then mixing with the general flock or onto the main pastures. So any questions on that section or not? Any questions are good. I'm not, no, okay. We'll continue. So I want to talk a little bit about grazing management, which is something we can do to obviously alternative to just relying on the drugs. So I think, you know, again, the caveat behind this is obviously this is very dependent on the individual situation in your flock, obviously. So what I'm going to do here is talk about principles, and then people's ability to apply them will obviously vary a lot in terms of their individual situation. But what we're trying to do is trying to prevent the buildup of these larvae, and then also preventing sheep having access to those larvae and ingesting large numbers of them. So when we think about what's happening at the flock, at the pasture level with these infective larvae on a pasture, there's this stylized diagram here where we have the months of the year, and this red line shows, you know, it's a stylized thing just to show what happens to the numbers of larvae on the pasture. A set stock pasture, which has just got sheep grazing, same group over the year on that pasture. And so what happens if we look at the egg counts of the ewes, which is this black dotted line, they peak around lambing time, and that's because of nutritional stress on the animals and effects on immunity. And so you get a lot of egg output from the ewes. And so the ewes themselves are a big source of eggs onto the pasture. And then those eggs will eventually develop to L3s, and eventually that will start to climb. And then once the lambs start grazing, they will then start to produce eggs and they will contribute. And of course, when these get to L3s, they get re-ingested by lambs and ewes. They develop to adults and the whole thing kind of multiplies. And so as you go through the year, these numbers build up. And so if animals are left on the same pasture or too long, then that's where you start to see problems. One question is, do these larvae survive from year to year on pastures? Well, we've got some studies from Eastern Canada on that, and the answer is no, Ammonchus contortus doesn't. But unfortunately, those other species do. And actually, we haven't done a study in sheep, but we've done a similar study in cattle here in Western Canada, got very similar results. So it's clearly applicable here. And so when you think about a pasture in the spring, and this is the number of larvae on that pasture, really some of these guys, not homuncus, but these other species will be there from the previous year if sheep have grazed on that pasture the previous year. But as it warms up, they will just gradually die off as they start to use up the energy reserves. They survive longer at cool temperatures and they gradually die off. And so if that pasture is not grazed, then by June, July, then all those things that have survived from the previous year will have gone. And so that when you're thinking about pasture in the beginning of the year, whether it's got any parasites on it or not, if it didn't have sheep on it the previous year, it won't. If it did, it might have some of these, but it won't have homuncus. And so again, I'm not going to go through it all again, but I've just, again, visually shown what I've just described about this build-up and the cycling through the sheep of these larvae in the pasture. Another wrinkle on this is that when these eggs pass out and they develop to L3s, they speed up in their development as it gets warmer. So they take much longer to go from eggs in the feces to infective larvae earlier on than they do later in the year. And then the other wrinkle is them migrating out of the feces onto the pasture depends on moisture. And so after rainfall, that will happen more and more quickly. So again, the point is it varies with the weather and the time of year in terms of the details. But it's notable that homuncus, which is this blood-feeding severe one, can get from an egg passed out to this guy, which is infective, in as little as 5 days under optimal conditions in this kind of time of year. So it can happen quite quickly. So when we think about rotational grazing, just one general point to make is that, it's good anyway if we divide pastures up and move animals along, not just for pasture management, but for parasite management, simply because most of the parasites in the deeper part of the pasture. And so if you took two pastures with the same number of larvae per hectare, these guys are ingesting way more on an overgrazed pasture than these guys. So simply moving animals and stopping them overgrazing in itself is a huge benefit. But on top of that, if you were to strip graze, for example, theoretically, you can literally manage parasites completely with these kind of systems. And again, I go back to the point, this is not possible for everybody, obviously, and I'm not suggesting it is, but by understanding this concept, it can be adapted at least partially to the individual management situations. So if you think about a situation where you are moving sheep from plots to plot every 2 weeks throughout the season, it doesn't give the larvae time to get from the egg to the infective larvae on the pasture and then infect the sheep. So if you keep doing that, you would stay ahead of it, and so there wouldn't be a source of infection for the sheep of any significant amount. Now, the thing is, these larvae do survive for a good number of months after they've been deposited. So what you don't want to do is do this and then move them back to one of the original pastures the same grazing season, because then you're putting them on a pasture with potentially lots of larvae there. So how you do this obviously can be adapted. The details depend on the stocking density in terms of, how quickly this will build up and the risk, initial pasture contamination, the species of worm, as I said, the time of year. So again, this time period here can be longer earlier in the season than later in the season, but then you've got to factor pasture growth into it as well. So there's lots of things here, but the simple fact that by moving sheep several times throughout the season can make a massive impact in reducing parasite burdens. And again, in itself, you may not be able to do it to the extent it would be the solution, but as part of a solution, it can be highly effective. There's also more interest these days in bioactive forages, and these are forages which are rich in condensed tannins, typically, and things like samphorian, trefoil and sulla are examples. And these do have real benefits, and they do have antiparasitic activity. There's a variety of trials been done, both in cattle and sheep, which show an effect. Problem is it's somewhat variable, and it's not a solution on itself, again, but it can help. And then there's obviously all sorts of issues about, managing these types of pastures in terms of their longevity and nutritional value and the rest of it. But it is something as part of an integrated program that and I know a few producers here have tried this. So I wanted to just finish off before we go into any questions with more of the blue skies piece in terms of where we're headed in the future and some of these alternative methods that you may have heard of and I get questions on, to really try and say kind of what works, what doesn't, to what extent. And so I just want to spend a little bit of time at the end on that now. So one of the things I've already talked about, so I've kind of covered that in terms of whether it works or not, these alternative forages. Another thing you might have heard of are these nematode trapping fungi, which are kind of neat idea and actually have been commercialized in Australia. So these are fungi which actually you feed the spores to the sheep, and there's a product that can be used to do that. It can be added to feed, and basically those spores pass through the sheep's gastrointestinal tract, and then basically the fungal hyphae grow in the fecal part, and they actually destroy the larvae in the fecal part, and so prevent the pasture contamination. And it does actually work moderately well. Again, it is commercialized in Australia. It's not available here, but I believe it is in the US. And so some people are using it there. And again, it's an adjunct rather than a complete solution, but it's certainly got some merit to it. The other you might have heard of are copper oxide needles, which are used more in the US. Copper is very toxic to homuncus. So, these copper oxide needle capsules are often used as a copper supplement and can also be used in helping homuncus control. Again, it's not something I like. They're very variable in their effectiveness. They do have some merit, but they're very variable. And if it's copper-rich pastures, there's a toxicity risk. Another one is a vaccine. You may have heard there's a vaccine against homuncus. Now I'm going to spend a couple of slides on that, so I'll come back to that, but it does have some merit. And then breeding for resistance to resilience is taken off in a number of countries and definitely has merit if people can incorporate parasite resistance and resilience into breeding programs. Two, which you may have heard of, which don't have merit, diatomaceous earth is these kind of fossilized particulate organisms, like a sandy material, which are actually used for pest control in insects, but they have no shown efficacy against roundworms at all. And so that's not something I would advise. And the kind of herbal remedies such as garlic and ginger and these things, there's no evidence I'm aware of any significant efficacy. So again, those are not things which I would incorporate into a worm control program. So I want to say a couple of words about the vaccine, because I've had a few questions about this over the last year or two. There is this vaccine, which again has been commercialized. It's sold in Australia. And there is a website for that. And if you just Google Barbavax, you can go to their website and look at the information. It's a neat idea. It's a great bit of science. The idea behind it is that the vaccine's based on these components of the worm's gut. So if you look at a worm in cross section, which is what this circle is, imagine a worm cut in cross section, you're looking at the end of it. That's its intestine. And there's these proteins on the surface of its gut, which they've purified and used to vaccinate animals with. And it's called a hidden antigen because in a normal infection, a sheep never sees that and never mounts an immune response to it. But if you inject it into a sheep, antibodies are produced to it. So when homuncus feeds on blood, those antibodies are present in the blood and destroy the worm's gut. So it's a really neat idea. And they've manufactured it. It's kind of neat in Australia where they do this and it was developed actually, I should say, by the Morden Institute in Scotland, and it's been commercialized in Western Australia. And basically they grow huge quantities of worms in sheep and harvest the guts, and then harvest this protein complex from the worms, and then basically produce the vaccine based on that. And it does reduce egg counts quite dramatically. This is from their website, where you can see these reduction in egg counts on different forms of vaccinated versus control co-grazed sheep. But the problem with it is, you need to administer this every 4 to 6 weeks, and that's because there's no natural boosting from field exposure. You get immunity and then it gradually declines. So because of that, it's kind of limited, or has been limited, to those places where they've run out of drug options and parasite problems are severe, that they kind of ran out of options essentially in some of these regions. And so the vaccine was a godsend in those areas. In somewhere like Canada, it's less clear where the benefit would be, and the cost and labor involved administering the vaccine may well outweigh the benefits. But there is some thoughts about using it in ewes. I know Paul Menzies in Guelph has talked about doing maybe some research on this. I've chatted to her about it, where you can imagine vaccinating ewes in the fall and again in the spring, and that might do enough to decline that egg production and contamination pasture from the ewes. And so it may have a role here, but at present it's not. I mean, it's possible to source it through import permits, but it's not something which I'm aware of anybody's used here yet in terms of Western Canada, but it's one to watch. So I thought I'd wrap up with that because I'm kind of out of time. I mean, the take-home message is this: we need to move away from this assuming drugs work and relying solely on them to this more integrated approach. And again, it's tailoring it to the individual farm situation. And so really thinking hard about what in these components can be done in your system, and also whether there's a need, because we see everything from producers who've used drugs when they don't need to use them, to others who are using them and not working, to others who should be maybe using them and they're not, etc. So it really does depend on the individual situation. And with that, I shall thank you for your attention, and also just thank again all the producers who've worked with us over the last few years, and the people who've worked on the projects. And please check out our website, because what we want to do with this website is it's got the basic framework now there, but we want to use it to continually update in terms of parasite control, keeping it relevant to what's relevant here versus as too many generic things. So thank you for listening, and I would really encourage questions if you can muster some.
Thank you so much, John, for your presentation. Like you said, we'll open it to the floor if there are any questions. You can unmute yourself, or you can send them in the chat box.
And I'm open to any type of question. I might not know the answer. I know some things about some of these topics in depth, and some of them not as much. So you're quite likely to catch me out with questions, but I encourage them if you've got some. I mean, one question I may put to you is how many people are using fecal egg counts themselves? Are there people who do that? If you do, you could just put a Y in the chat box, and it'd be interesting to see. Or is nobody using them? So we shouldn't deworm in the winter. No, absolutely right. Actually, that's another good point we probably didn't mention. So the transmission really all occurs in the summer, in the winter. So when the eggs are passing out in the feces, once you get past, probably it depends obviously on the year, but once you get past end of September, most of those eggs will just die because the eggs themselves don't like cold temperatures. The larvae on the pasture will survive, but the eggs won't. So in other words, they won't be picking up, and they won't be picking up too much in the winter, and they won't be contaminating the pastures because of what I've just said. So any treatment in the winter is kind of wasted, really. So the time we treat with drug is used in the spring, because we're trying to prevent that pasture contamination. And what I'm trying to encourage people to do is do that, but based on experience of fecal egg counts and only treating part of the group of ewes. And then the lambs may need treating later in the grazing season. So we're talking July, August, September, depending on the situation. But again, triggered by monitoring, and again, not all the lambs, but some of the lambs. So you're treating spring ewes, lambs in the later part of the season, and that's the kind of timing, but again, only a proportion. So somebody said we use fecals and send them away to our vet. Yeah, no, that's good. I'd be provocative and say if people aren't using fecals, you should question why. If they're using drugs and not fecals, I think we should be trying to move away from that. Cost, I know, is an issue, but when you pool them, it really is quite cheap, right? So it's not, cost isn't in itself a huge issue, but I do realize time and effort is a factor for people, and it's easy for me to say, but I think it's a relatively simple thing to do. I mean, collecting fecals is very simple. I mean, you can gather sheep and then just pick up fresh feces from the ground, and so it can be done quite quickly and easily. So question, if keeping sheep off for a year, can you take hay off that is not contaminated? Yeah, so yeah, absolutely. So if you're making silage or hay, that's not a risk. So the parasites won't survive hay treatment or silage treatment. So the hay or the silage itself is not a risk. It's fresh pasture and grazing. So it comes back to this indoor thing, is that there'll be no transmission indoors, either from the bedding or the feed. That won't happen. So when you're thinking about either making forage or if you're thinking about sheep indoors, it's all about when they have access directly to pasture. What's the next question? Let me have a look. We rear indoors and haven't know them yet. We definitely use fecals before administering the wormers. Yeah, absolutely. No, that's good. And if you're rearing indoors, as I say, if it's entirely indoors, then there will not be worms. You might have problem with coccidia, which is a different parasite. That does cause problems indoors, but roundworms don't. So this is really about grazing animals. But having said that, yeah, before moving on to any grazing, it's always good just to take a fecal and checking, because that's the risk point of when you move on to a pasture. And if it's a pasture that hasn't been grazed for a long time, any sheep that go on there having been treated with drug will be seeding that pasture with resistant worms. And there's low refugia, and that'll be a strong selection for resistance. So those are the checkpoints whenever moving animals onto a clean pasture. Taking a fecal egg count, particularly after any drug treatments, is very important. Any other questions or anything anybody disagree with? Or I'm actually quite, what's useful to me is if people say, well, that's interesting, but it's impractical, or I'm always happy to hear those comments, because it's, what I'm trying to do is provide a framework of good practice, but clearly some things are easier than others for people to do.
We'll give it another minute or so to see if there's any additional questions that are coming through.
Yeah.
Well, it doesn't appear that there's any other questions that are coming through here, John. So thank you so much to you for presenting to us this evening, and to all of our participants who joined us this evening. This will conclude the webinar. I will be sending out an email to everyone who registered for this evening session that will include a link to where you can access the webinar recording.
I've got one more question.
I saw that just came through, so this will be the last one.
Did you say very dry conditions like this? Yeah, absolutely. Yeah. So these parasites like moisture. So again, these are good things that kind of forgot to highlight. Yeah. So when you get wet weather, you tend to get more parasites, and there's a couple of reasons for that. Is one, the larvae survive better on the pasture. They don't like dry conditions, tend to dry off, but also them escaping from the fecal pat, so they come out in wet weather from the fecal pat onto the grass. So if you get really drought-like conditions, it's going to generally be a low parasite year, and wetter years are going to be a higher parasite year. And another thing to mention of that, of course, if you treat with drug in a very dry year, it's a good example actually of you probably didn't need, or you may well not have needed it, and you're also selecting for resistance because those few parasites that are there are not competed out by things on the pasture, and there's that diffusion thing again. So dry conditions, you really should be questioning whether you need to treat or not, and then you'll only know by monitoring. So thank you, that was a good question.
All right, well, I think we're good now. So thank you once again, John, for this evening. It's greatly appreciate the time that you spent with our producers this evening.
No, I enjoyed it. Thank you. Thank you for listening. I'm always happy to talk about parasites. So
Well, have a good night, everybody.
Okay, thank you.